Okay, so we are continuing our tour of the cell. Where we stopped previously, we had ended up starting from our outside of our cell and working our way towards the inside of the cell, right? So we had looked at the plasma membrane, we had looked at some of the inside of the cell. We talked a bit about the cytoplasm, the intracellular fluid, etc. And as we were talking about the plasma membrane, we said that this is a fluid mosaic. Remember, we said that it controls movement into and out of the cell. So remember, with the fluid mosaic, moving all the time, different components to it as well. So that's a key thing to know about the plasma membrane. You need to know the different molecules that make it up and why those molecules are there. We're talking about different types of proteins, phospholipids, glycolipids, cholesterol, sugars that identify it, all kinds of things like that. Okay, the last topic where we were is we were also looking at the different forces, if you will, or different ways that things can go across the membrane, so membrane transport. We finished up with passive transport, doesn't require energy, and now we're moving on to active transport. So active transport, there's two different types. There is what's called active transport itself, and then there is what's called vesicular transport. So both of them are going to require ATP to move across the plasma membrane, but for some reason, whatever's moving across, if you will, doesn't want to go that way. Remember, with passive transport, it's like a ball rolling downhill. Doesn't take any energy. It goes from high to low. And in this case, maybe that ball is basically too large to be able to go through. In this case, go through channels, or it's not lipid soluble. Remember, this is a phospholipid layer, and so it needs some sort of a transport. So we're going to wrap it in a bag, if you will, and get it across that way, a bag like a vesicle. Or it's going against its gradient instead. So if it's going against its gradient, it's like a ball going uphill, right? That's going to take some energy to make that happen. Okay, so if we're going to be doing that, we need those proteins to be able to move these solutes across. And so they're going to bind with that particular substance for a while, and then they're going to get it across, and then they're going to switch their conformation, they're going to change their shape, and they're going to be able to potentially grab another one. Now, when they grab a substance to move it across, it could actually be more than one substance. And if we have two different substances and they're going opposite directions, that's called an antiporter. So the sodium-potassium pump is an antiporter. So sodium is going out of the cell and potassium is going into the cell. And then symporters, they're going to be bringing two different substances in the same direction. So there is a sodium glucose transport mechanism where both of those things basically go into the cell. So there are different ways where we can actually move things across, but they're going to involve those proteins that are embedded within the plasma membrane. So like we said, in these cases, what we're doing is we're generally moving these things against their concentration gradient, having them go uphill, and that's going to take energy. So in terms of active transport, first of those two categories, there's two different types of active transport. Primary active transport, where we have to burn ATP or hydrolyze ATP in order to give the energy to move these things across. So for instance, again, sodium-potassium pumps. We have to take an ATP and hydrolyze it, break it apart, and utilize that energy, decomposition reaction, big thing down to small things, in order to give it the energy to actually turn and pump these things across. Same thing with calcium pumps and hydrogen pumps, etc. Now, secondary active transport is a little, I wouldn't say more confusing, but it uses energy but not directly. So in this case, what happens is there is, in certain types of cells, a gradient. So when we talk about energy production, ATP production, later on, we talk about how we can produce ATP, and it has to do with a hydrogen or proton gradient. And basically, it's kind of like a waterfall, if you will. Sometimes you see waterfalls and they have a turbine associated with it. So as the water goes through, or goes through turbines associated with a dam, as the water moves through, according to its gradient, from one side of the dam where there's a lot to another side of the dam, it actually can turn or provide the energy to turn this mechanism, a turbine, or in this case, like a pump. So that also happens in cells. So if we have a bunch of ions on one side of the membrane and not as much on the other side, if we allow them to go and diffuse the way they naturally would, it can actually provide the energy to turn a pump. So in a way, the reason that's called secondary active transport is we started out with what's called a proton pump in this case, or a hydrogen ion pump, where we are pushing those ions and piling them up outside of the membrane. So that took energy originally, but once we allow those ions to come back and flow down their gradient, now we're utilizing that movement in order to produce the energy for something else, like transporting something else. So in any case, secondary active transport, it initially used ATP, but what's really providing the mechanism for this type of movement is actually the movement of these ions that have been stacked on one side of the membrane and are now going back again. Okay. So, also, when we talk about these membranes, and we're talking about moving things across the membrane, there's a key thing that's going to come up as we get into these different systems, in that there are certain types of cells that use these distributions of ions on either side of the membrane for a particular function. So for instance, our excitable cells, like muscle cells and neuron cells, etc., they will actually have what's called a membrane potential, where they have piled up a bunch of charged particles, ions, right? Cations, anions, things like that, charged particles, even charged proteins on one side of the membrane or the other. And when these cells get stimulated, they'll allow these different ions to flow and diffuse one way or the other. And as we move charged particles on one side of the membrane or the other, that actually changes what's called the membrane potential. So it's an electrochemical gradient. And that's going to be important in terms of conducting a signal. So much like charged particles, if you have an electrical outlet or something like that, you have charged particles there that are moving, and maybe they're moving through the wires that you have, right? And so in a way, that's what's happening in these different types of excitable cells as well. They're moving charged particles that allows them to send signals or transmit signals, etc. So we'll see that coming up. Here's a little movie that shows something about that as well. We'll see that in again, mostly we'll look at it as it pertains to these excitable cells, so we'll get to it in muscular system and nervous system, but we do talk a little bit about it here because most all cells do have some, like we said, distribution of these charged particles on one side or another. That's the role of the plasma membrane. So here we can see our example of two examples of different types of transport, right? Primary active transport, where we hydrolyze an ATP in order to pump sodium out and have potassium come in. And then secondary active transport, where we've actually pumped the sodium out and, sorry about that, pump the sodium out, and now the sodium wants to come back. And so if we can allow the sodium to come down its gradient, this symporter, which transports sodium ions and glucose, will bring not only sodium but glucose back in. So the glucose kind of hitchhikes a ride in with the flowing sodium to bring glucose into the cell, which is very critical because we know that that is going to provide the chemical energy for the cell to do its work. Okay. So other things, we talked about active transport. There's also vesicular transport. So a vesicle is a bag. And so it's it's just like the plasma membrane for the cell, but it's much smaller. So we've created, almost pinched off a section of the plasma membrane to create a bag where they can then transport this substance across. So much like if you are in a grocery store or leaving a grocery store, it's much more convenient to transport a bunch of different substances using a bag or a grocery cart where you can package them all together. Now, when we're moving these things, if we're moving them into the cell, we call that endocytosis. And if we're moving it out of the cell, we call it exocytosis. Now, endocytosis, there's 3 different types we'll see coming up. And then exocytosis, that is exiting the cell. That's when we are transporting these things. This could be substances that are going to be used in other types of cells. It could be we just want to get it out of the cell because it's a waste product, etc. Endocytosis, we'll find, is when we have a use for that particular item. And then we also have transcytosis, where it is being transported through the cell. So for instance, the cells that line, the epithelial cells that line the digestive system, when we're absorbing nutrients after we've digested them using those decomposition reactions, see, chemistry, they're going to come through the apical surface, the top surface of those lining cells. And then it's going to transport through the cell, and then it's going to go through the basal surface and into the bloodstream. So it has to transport all the way through. Also, we have some trafficking in terms of not going into the cell or out of the cell or both, but within the cell as well. So that's called vesicular trafficking. So this figure that we see here just kind of shows us that. And you may notice as you go through the lectures, oftentimes I will focus on the figures from the text, or I bring them in from other sources, etc. To me, right, the phrase is a picture is worth 1,000 words, and this one actually has almost 1,000 words on it. But I think it's much better to have something that you can visualize, particularly on a subcellular level, but on any level, it's better when we have a picture instead. So if we have a picture like this, then I'll talk about the picture itself, but the previous slides that lead up to it where it has all the text, that's basically for you to read over. And so if you're reviewing or looking at the lecture notes beforehand, that way you can get an idea about what I'm going to be talking about. When you're looking through them afterwards and you're studying for the test, it gives you that text of critical information that you need to know. But for the most part, I tend to focus on the figures themselves when I'm actually doing the lecturing. Okay, so like we said, different types of vesicular transport. When we look at it, here we see endocytosis, right? Coming into the cell, we've grabbed something and we're bringing it into the cell here. On certain types of cells, we may actually digest it in order to break it down. We can see some transcytosis here where we're exiting the cell, doing exocytosis on the other side. So you can see where these different types of vesicles kind of arrive though. We get a little pocket of the plasma membrane and then it pinches off and encircles that particular substance for transport. So it's kind of like you load the bag, and then you tie the bag at the top, and now you have a nice little package. Okay, so in any case, for endocytosis, we have a few different types. One's called phagocytosis, so phago means eat. And so with this one, these are for large particles, right? So if you are ingesting that In-N-Out burger, that's going to be a large particle. On a cellular level, this is also cell eating. It's grabbing it, and it's going to bring it into a bag, if you will, that is basically called a phagosome, right? An eating little bag. So we have phagocytosis, we have cell eat, we have penocytosis, which is cell drink. So this is going to include small particles or solutes that we see, but it also includes some of the extracellular fluid. So that's why we call it cell drink. And then receptor-mediated endocytosis, this one's very, very specific. So we can see these little protein receptors in the membrane, and as they bind this particular specific substance, once we have a bunch of them bound to the receptors, we're going to fold that plasma membrane in, and we're going to bring all those in at once. So these are only bringing in one particular thing. Cell drink, there could be all kinds of things coming in from that extracellular fluid. And this just shows us a little bit of exocytosis. So again, we have a vesicle, and in this case they call it a secretory or secretory vessel because the substance is going to be secreted into the extracellular fluid. But it's kind of the reverse process of what we saw for endocytosis, but we have these docking proteins that are here, where as we move that vesicle towards the plasma membrane, it's actually going to then be transported to go down and dock there and then open up and release that particular substance. Okay? Okay, so like we said, these movements of substances across the plasma membrane is going to be very important for eating and drinking of the cell, but also on a smaller level, the movement of charged particles is also going to be important in all cells, but particularly what types of cells? Excitable cells. Muscle cells, neurons, etc. So that membrane transport that we're talking about produces what we call resting membrane potential. So again, this is the separation of charged particles on either side of the membrane. And so when you look at this, right, voltage, that reminds you, like we were talking about electrical wires and stuff like that, right? Voltage is actually where we are seeing that separation in electrical charge, just like if you're talking about, you know, a 9-volt battery, or you're talking about 120 volt, right, outlet or whatever. So these cells where there's an unequal distribution of charge on the inside and the outside are called polarized. And a lot of these different cells for what's called a resting membrane potential, where they're resting and they're not doing anything actively, it sits at about -50 to -100 millivolts. See, even the units, right? Volts, but this is millivolts, right? 1/1000th. So -50 to -100. And what that means is, with a negative sign, that means the inside of the cell is more negative relative to the outside of the cell. So that means that we could have a bunch of positively charged things piled up outside the cell compared to inside the cell. It could mean that we have more negatively charged things on the inside of the cell relative to the outside of the cell. Both positive and negatively charged ions and molecules will contribute to this resting membrane potential. And one of the most important player in resting membrane potential is this that we see here. So what do we know about that? What is that? So first off, we know it's an ion, right? Is it a cation or an anion? So that's a cation, right? Cation is where we have that plus, tells us right there, the plus charge. And K, so it comes from the Latin kalemia, so this is actually potassium. So even though potassium for us starts with a P, the K is the symbol that we have. But what we have is that the potassium that we see here basically is moving across the plasma membrane through what we call leak channels that are open all the time. So the distribution of potassium and sodium contributes some as well, helps to make this resting membrane potential. And potassium is the main one that contributes to it. So what we find is potassium is constantly leaking out of the cell down its concentration gradient, and as we lose those positive things out of the cell, it makes the inside more and more negative. So it sets up an electrochemical gradient. Like we said, sodium, right, natrium, it's also a positively charged ion, and in fact, that sodium contributes some as well. And sodium is high outside the cell and low inside the cell because of a sodium-potassium pump, and so it wants to diffuse back into the cell. And so rather than being at about -90 millivolts, if we were just looking at potassium, due to the sodium, the positively charged sodium leaking back into the cell, that makes a resting membrane potential, on average for a lot of cells, at about -70 millivolts. So this is something that we'll see as we continue on. So this is the figure like I was referencing, where again we see that distribution. So I got my multiple screens going, pointing all over the place. We see distribution where it's higher concentration of sodium outside the cell compared to inside. It's higher concentration of potassium inside the cell relative to outside. But with these leak channels, by losing those potassium ions all the time as they're leaking through these proteins, that makes the inside of the cell slightly more negative compared to the outside of the cell. And we also said that the active transport of sodium and potassium to maintain high sodium on the outside and high potassium on the inside is due to a sodium-potassium pump, where it's constantly switching 3 for 2, 3 for 2, 3 for 2. 3 sodium go out, 2 potassium come back in. Okay, so resting membrane potential, all cells have a resting membrane potential, but again, we'll talk more details about that when we start talking about cardiovascular system and muscular system and nervous system, these cells that make it up and that have this excitable nature to them, where they actually use these changes in membrane potential. Okay, so that's a little bit more about finishing up this idea of the plasma membrane and how we move things across it. There's just a couple other things that have to do with that plasma membrane. Do remember that we do have some proteins that work to adhere cells together, like in a tissue, as well as these are the eyes and ears, if you will, of the cell. So they're able to receive messages as well using some of these proteins that are called receptors. So when we have these types of proteins there, the receptors, again, serves like ears and eyes. They're actually being able to communicate with the surrounding environment, right? So they can be responsive to it. So they can respond to it by being attached to another cell and this contact signaling. So as the cell grows, if it starts to butt up against another cell, it will start to get information about that because these proteins are kind of getting squished, if you will. And then also chemical signaling, right? Hormones and neurotransmitters, those are going to reach target cells and be bound to these particular proteins. This example that we see here is a cascade system, what's called a second messenger system, where you have some sort of a chemical messenger like a ligand, binds with an initial receptor that's embedded within the plasma membrane, and in like a domino effect, it sets off a series of events that then gets the signal into the cell. And also, you get a massive response, a whole bunch of amplification where the cell responds by cranking out a bunch of enzymes of its own or hormones of its own, etc. All right, so that finishes up the plasma membrane. You wouldn't have thought that there was so much detail about just that little line that we were talking about before, that we were thinking of before, but you can see it's kind of complex. So our next step is we will move into the inside of the cell coming up.